Hello everyone, I am going to record the session of molecular docking. So today first of all I will be typing the PubChem where I can download my ligand in stdf format. So I just mentioned here a PubChem on the web searcher. Just click here PubChem. So now you can mention the name of your drug on which you are going to perform the molecular docking. So in class we have taken zidovudine as a drug. So the same I am also mentioning here. Zidovudine. So the search is ongoing. Generally it takes 30 seconds or 2 seconds or not more than that, but sometimes it takes more than a minute just because of the network and all that problem. So come to the second one. So okay, now the things is also being displayed. So as you can see, this is the zidovudine. This is the molecular weight, which is the 267.24 gram per mole, and always try to take the compound which is less than 500 gram per mole all Dalton, because then it only follow the Lipinski rule of 5. Otherwise it do not follows the Lipinski rule of 5, and you have to drop this drug if you're going to provide the human medication, all that. Okay, so make sure whatever the drug that you are going to take for docking, it must be less than 500 gram per mole if you are going to work on a human disease or provide human oral bioavailability drug like that. Okay, so this is the canonical smiles, where which you can see this is the one other way of representing this compound is by using the canonical smiles in Campbell keys and in Campbell's like this description as well. And this compound is submitted here on 24 June 2005. This is the CID, or you can say as an identifier, which you can help of which you can identify the compound. If you mention 35370 in the search box, you will again get the same result as you will mention the zidovudine. Okay, so each compound has a unique CID in PubChem database. So make sure you have to be clear about these things. Either you can mention the name of that compounder, or if you can know the CID of that compound, just generally mention the CID. Okay, not anything else. Or if you can also search using the canonical smiles that you know. Okay, canonical smile is nothing but the molecular formulas. Molecular formula matlab like in this way. Okay, where everything is covered. This is a descriptive molecular formula. That's why it is termed as a canonical smiles. So now I click on this part. I have to click here download, then click on SDF. As you can see right now, this file is downloaded. Okay, now coming to the next one. Now for protein, we have to go to the PDB database. So just open a new tab. Just mention here PDB. Enter. Now just click here. Okay, now you have to mention the protein name that you want to work. Someone has suggested in class RNA dependent RNA polymerase. So I am mentioning the same protein here. RNA dependent RNA polymerase. Enter. So actually it is a viral protein, like especially in all the virus part, this protein is present. Okay, and causing some toxicity, or you can say as a disease like that. So let's see in which virus the result is being shown. So the search is ongoing. Meanwhile, just come to the third one, which is castp, that is used for active site prediction. Just I mention here castp, enter. Okay, now yeah, I have to upload my protein on which I am going to work. Like click on calculation. On clicking the calculation, you have to now upload your protein. Here is the option to upload your protein. So let's download the protein from the PDB. Still the search is ongoing. Let me just wait for a second. And after that, once the proteins and drug is downloaded, you have to do one more thing. You have to create a folder like with name of biotech on. So inside the biotech on, you have to create two more folders. One is ligand, second is protein. In ligands, you have to paste your drug file in SDF format, and in protein folder you have to paste your protein. So yesterday I downloaded for the class, which is 5IQC is a protein. So I'm not doing it again, like the thing is ongoing. Same thing, I have also downloaded a drug once again, this time as well. Like this is the zidovudine. So one, you can also rename this drug as a zidovudine, or you can leave it as well, like just leave it as that. Okay, now come to again this one. The search is still ongoing. It might take couple of minutes more. Okay, now meanwhile let me show you how to use the castp. So I have to upload my protein. So yesterday I downloaded a protein. I will upload the same protein here once again. Okay, and now click here submit for the active site prediction. I explain you what is active site in class. If you don't understood, today I will again repeat the class, so don't worry about it. So this web server is quite not working proper. Sometimes it takes a quite more time for predicting the active sites, and sometimes it takes very less time for predicting the active site. You have to click on submit twice thrice, okay, because it is not functional as this moment at this moment. Okay, so as you can see, yesterday I submitted the protein and I got a result like this, where I can get to know what are the pocket IDs and which chains are there, what the sequence IDs, their amino acids, and the number of atoms. Okay, so come to the this one, you have to click here download the active like castp result. So once it downloaded, it will come like this. A folder will get downloaded and it will look like that. Let me show you. Like this. This is the castp result. Okay, just click here. You can will see like 6 files there, and you require only one file, which is POC file. Okay, now double click here and like to open, okay, this file. And most of the time it will open with any other thing, like any other tool, but try to open with notepad. Okay, now copy all the content of this file, the POC file. Remember, I'm again repeating, it's POC file. Okay, out of all this file you require only POC file. I have copied the content of POC file. Now I will open the Excel sheet. I will paste there the content and find out the active sites from there. Okay, yeah, this time I also downloaded, which is already downloaded, but don't worry, I'll go with the previous one. So now the Excel sheet is opening. I have opened a new blank workbook. Come to here A, paste all your data. Okay, so this is all the data of POC file that I have copied from the castp, okay, from castp result. Now select A, go to data, just click here text to column. Here you can directly click finish. So all data is now distributed in these columns. Now again what you have to do, you have to remove, you have to take only two things. One is this column which I'm filling with yellow color, and second is this one. Rest all are like not important for the analysis part, okay, just remove it. So selecting one, each one by one, I am removing all this. Okay, now I'm left with these two like columns. Let me unfill the color. Same goes for this one as well. Now again I will select column A, go on data, click here remove duplicates, expand the selection, remove duplicates. So just click only one column at a time, like column A only, not column B. Click here OK. So as you can see, 1277 duplicates were there, which is right removed. And as of now, only 21 unique values is left. 20 or 21, okay? So these are the unique, active residues. So this is the important thing which you required, okay? Now save this file, save. Okay, you can save it. So it requires the name. Wherever you want to save it, you can save it, okay? I don't want to save. I just want the content. Okay, I just copied the content and the folder in which you are going like working. Like in Windows, say I told you to make a folder named biotech ligand protein. Just make one more a file here, text document file. Just name here castp. Enter. Okay. Now here you have to paste the content. Just save it. Okay. Yes. Okay. Now this information is right now saved. That's all for today. Now in today's class, or like we will see the further steps, okay, how I can use this information for predicting, the grid file and all that. And also to explore more about the protein, I will be showing the Discovery Studio tools, like how that is very very helpful for getting no binding pockets, and more and more exploration can be done using the Discovery Studio, which I will show you in today class. So keep follow this video. I hope it clears all your doubt. If you have any other doubt, please let me know in the